I'm right-handed, so I probably stand here so I can face you and point. So I'll talk about multi-photon imaging for going deeper in the mouse brain. So how many people are familiar with multi-photon microscopy here? Oh, all very familiar. So I can skip the beginning part very quickly. So first acknowledgement, this work is done by roughly half my labs. 2 students did the most of the work and 2 postdocs. And my lab also do a lot of the fibers and laser development. It turns out to be very synergistic. But today I just talk about an imaging effort. Collaborators, a lot of them help us in different domains. For example, Chris and Izumi help a lot on the animal preparation for us. And a lot of people give me ideas like David Tank in Princeton and where's the adaptive optics? Naji on the adaptive optics. And we work with several companies, mostly on the laser development, including now Coherent. We also work together, try to get the best laser for doing deep imaging. Funding, mostly now transitioning to NIH, started with telecom, but now mostly funded by NIH, but also with the Defense Department and the Intelligence Agency, some foundation to support our work. So the motivation is very simple. We want to go deep in the brain. It's really no-brainer here, right? You look at the mouse brain, 1 cubic centimeter. This is roughly the best 2-photon microscopy could do, this yellow box. Maybe on a good day you go, say, 600 to 700 microns deep. That's a good 2-photon performance. A few hundred microns across with hundreds of neurons. That's roughly 2-photon performance. So about 10 years ago, I transitioning from telecom back into imaging. I was thinking, can we push this a little bit deeper, say go twice as deep? Can we do that? And of course, can we also image much wider area to cover many more neurons? Say, can you cover the entire cortical column and things like this? So that was our goal. We start to drive this. And to quantify, in some sense, we still try to achieve this goal, is try to do volume rate of 10 hertz, spatial resolution submicron, at least 1 millimeter deep, and at least 1 cubic millimeter volume. If we can do this, I think we can answer a lot of interesting neuroscience questions. So it's still a major scientific challenge, and I hope I can show you that it becomes an engineering problem. We have the tool to do this, just how to make it happen. And if the brain is transparent, we know, like a baby zebrafish, life is much simpler. You do light sheet microscopy, for example, you can look at the entire brain activity, right? Tens of thousands of neurons. But when the brain is not transparent, you're in trouble because we know cameras does not work. If you go out in a dense, foggy weather, you take a photo, you know you couldn't see much because scattering. If somebody can figure this one out, how to do parallel data acquisition with a camera, that would be a major breakthrough. And we had to try that, and nothing works out very well yet. So very simple, cell phone camera. I put a lens paper in the front. You can see that photons reach your camera. You can still see this is not dark. Photons are there, but the information the photons carry is lost. And that is my office in Ithaca, New York, right? Photons are there, but no information. You look at the mouse brain in the cranial window, maybe not you probably have seen. It's the same thing, right? You have to think about looking through a thick piece of tofu, a thick piece of cheese. It's very hard to imagine to see a single neuron underneath, say, a slice of cheese on the top. So that's what we have to overcome. The technology we use is called multi-photon microscopy, started as a 2-photon microscopy by Winfried Denk and Walt Webb in 1990. And I joined Webb's group as a beginning graduate student in 1992. So this is always, I wish I'm a couple years older so I can join on this year. That's the only time you wish you're a few years older than who you are. So brief introduction, very simple idea. A fluorescent molecule typically absorbs blues emitting the green, fluorescence process. For 2-photon process, just putting 2 red photons, combine the energy, making the same molecular transition. And in general, that's the exactly same fluorescence process. And the only difficulty you have to overcome, in fact, we have to always overcome this for any nonlinear process, is that you look at the cross-section of 1-photon and 2-photon processes. Essentially, the probability of having 1-photon transition or 2-photon transition. You can see the probability for 2-photon excitation. Okay, the units is always very funny. It's a historical reasons. But you just look at the number. The number is much, much smaller than 1-photon transition. So that's always you have something to keep in mind. You do nonlinear optics, the probability is always very small. What you need is a perfect laser for that. So we use femtosecond laser. So the laser is mostly off. In fact, 1 part per million typically is on. Vast majority of time, it's 99.99% is off. But when it's on, it's extremely bright. By using lasers like this, we can actually make such a small sort of a probability into a reasonable biological contrast. So 2 photons are useful in that regard with a laser like this. So for a fluorescing molecule, you can see blue photon in, green photon out. That's fluorescing, FDA approved. You can drink if you want, right? Blue in, green out. And 2 photon, you can see, very trivial. Put the red photons in, combine the energy, exactly the same green fluorescence coming out. Now, because 2 photon is needed for 1 molecular transition, then the signal for 2 photon excited fluorescence goes as intensity squared. Okay, it's A plus A equal to 2A, kind of a chemical reaction in a sense. All right? So this nonlinearity turns out to be extremely useful. And the reason is that with nonlinear excitation, you can define a 3-dimensionally confined focal volume. You can see that fluorescing solution, fluorescing in water, 1 photon excitation, blue light, focal point, we can see lots of fluorescence above and below. For 2 photon excitation, because it's intensity square dependence, you can see that only the focal point gives you excitation. So this actually is the slide that convinced me to join Webb's group, literally 25 years ago. And the logic is still exactly the same. This nonlinearity, the nonlinear confinement, is essential for imaging deep in scattering tissue. And of course, you raster scan this point, you form a 3-dimensional image. That's how it's working. So the reason is, let's take a look at why 2 photon can go really deep in the tissue. You look at a confocal microscopy as a comparison. You excite fluorescence at the focal volume. For confocal to work, you have to re-image the emission onto a confocal pinhole. Now that works really well without any fog in between, without scattering. So you can image a point to a point. Imagine emission fluorescence gets scattered on the way back to the detection pinhole. Now you can see that's like through dense fog again. Instead of having a very nicely defined point for the fluorescence, you have now a large extended illumination volume. Now you can see the pinhole is not very useful here, right? It cuts away all the photons you actually really want. You go to 2 photon because we define an excitation point. That means we define the origin of my fluorescence. So in that case, we put as big a detector as we can find, typically like a centimeter. You collect all the light coming back. Now that is good because nothing is wasted over here. Now in addition, as I mentioned, for nonlinear excitation, we use lower energy photons. That means they're longer wavelength. Longer wavelengths typically reduces the impact of scattering. And that's why the sky is blue, sunset is red, right? Scattering is reduced as you go to longer and longer wavelengths. So if you look at the advantage of nonlinear microscopy is 2. One is nonlinear confinement. This issue gives you about 100 times improvement in signal collection, where they go deep. Deep means, say, 3 or 4 scattering lengths into tissue. And the second issue is this longer wavelength. And if you go again, a few scattering lengths deep, you still get about a factor of 100. So you combine them, you can see that's why it works so well. 10,000 times improvement comparing with, say, typical confocal microscopy in the blue wavelength. Okay, so nonlinear confinement and longer wavelength. And just to emphasize, this is the key, the ability to form a sharp focus in 3D. That's all it matters if you want to go deep into tissue. Once you can do that, you have penetration depth. So what's limiting 2 photon then, right? Okay, 2 photon is great, and we talked about it, but why can't I go through the entire mouse brain or even the human brain? What's the problem? The biggest problem is tissue scattering, as we know. Imagine a photon coming in, aimed at the focal spot. Distance, I give you a scale, right? You want to go, say, a millimeter deep. The focal spot typically is half a micron. So that means this is 1,000 microns and this is half a micron. You can imagine photon come in, try to reach this point. It's like what? It's like a sniper trying to shoot a human being from 1 kilometer away. Right? It's the same scale. Now imagine a photon coming in gets scattered along the way. Scattering means it will slightly deviate from its ballistic trajectory. When that happens, its chance of reaching here is 0. Imagine a sniper shooting a guy 1,000 yards away. Of course, the bullets hit something between, you have no chance, right? So that's the biggest problem. That means the excitation photons, the number of excitation photons at the focal volume goes down exponentially as a function of depth. So the exponential decay of the signal is really the biggest issue. And second, okay, just to emphasize this, it's experimentally proven. We image the vasculature, look at the fluorescence brightness as a function of imaging depth. It's a semi-log plot, log scale on a vertical axis. You can see the fluorescence goes down linearly in this semi-log plot. Of course, linear in semi-log plot means exponential decay, right? You can clearly see the signal goes down dramatically. When you go this deep, you go down by 5 order of magnitude. It's a huge penalty for going deep into tissue. And second, the brain is not a piece of optical glass, right? That'd be nice, but it's not, right? So you have optical aberration because you can see there's stuff in the brain that's different index. You have heterogeneity, and that causes optical aberration. What that means is that your signal will be decreased because you can't focus really well. And not only that, your resolution gets poorer. You can't resolve as well. So both are bad stuff. And lastly, even if you got signal this deep generated, you still have to propagate back up. It's all in epi, right? You do in vivo imaging, typically do epi detection. There's not much choice. You can detect in the bottom, right? So, and the signal absorption through the tissue, the fluorescence absorption by the tissue becomes significant. If you quantify this, about 5 order of magnitude, about 10 times over here, maybe 5 to 10 times for the last issue, okay? So you want the imaging deep, you really have to get those factors back or as small as possible. Otherwise, you're suffering 10 million times if you want to go millimeter deep. Now, that's very difficult to do experiment. So what's our strategy? Very simple imagination, right? You start to think. We first think about why 2-photon works so well. I just mentioned there's only 2 reasons 2-photon works so well. That is nonlinear confinement and longer wavelength. That's the reason. If you want to imagine what's a new idea, the simplest solution will be we push it to longer wavelength and we add a photon to it. So very simple logic, right? It takes literally, you can think it's very simple, a little bit crazy, but it really works. And if you put a time scale on this, it takes 1 minute to come up with the idea, literally 1 minute all you need. It takes 1 hour to show that it won't work, so you'll give up. It takes a couple of days to think it might actually work. And it takes 3 years to make it work. That's a PhD thesis. That's roughly what it is, right? So you need a little bit of quantitation to understand the 1 minute, 1 hour, and 1 day issue. So why it's the case. So why it's a little bit worrisome to go to long wavelength and 3-photon imaging. The first issue is water absorption. This plot really worries people. This is the water absorption on log scale. So each notch is a factor of 10. Now, typical 2-photon with a Tarsapha wavelength is sitting over here. You can see water absorption is very small. You go to long wavelength here, longer wavelength here, you can see the water absorption goes up dramatically by 1 to 2 orders of magnitude. That worries people. It's like you're absorbing so much, you're increasing the absorption so much, how can you make it work? You cook the tissue before you can image the tissue. The second issue, as I just mentioned, 2-photon cross-section is already very small. Now we're going to go to 3-photon excitation. The cross-section is really small. Look at the number here, right? 1 photon, 2 photon, 3 photon. The number is really almost non-realistic. And these are the 2 reasons actually we got 2 grant applications rejected in the very beginning, about 2010. People say it won't work. You heat the tissue and the cross-section is just too small. It won't work. But you need quantitation for that because, first of all, you do in vivo imaging, right? In addition to water, you also have blood. It's hard to get rid of blood if you want to image the mouse alive, right? If you look at the blood absorption, really dominates in a short wavelength. If you put it together, this is physiological blood content in the brain. About 10% of volume in the brain is about blood. You can see, now you look at this, the absorption difference between typical Tarsapha wavelengths and the 2-photon wavelength and these long wavelengths, the difference is much smaller because blood absorbs in a short wavelength. So first of all, the difference is not that big in the absorption case. And second, you really win a whole lot by reducing scattering as you go to longer and longer wavelengths. These are called scattering coefficient. The smaller the value, the lower the scattering. So you can see by going to longer wavelength, So from 800 to 1300, you're down by a factor of 2. You go to 1700, you're down by a factor of 3 to 4. So you really win in this case. So if you put it together, okay? Absorption, Beer's law, decay. Scattering, also exponential decay. You combine those things together, is multiplication. You sum up the exponent. You can define a new length scale. We call it effective attenuation length, essentially, is combining absorption and scattering, right? You put it together, you find it looks like this. These peaks are peaks of water absorption. The general trend follows this scattering curve. You can see now 800 is not very good. That's typical Thai sci-fi wavelength. 1300 and the 1700 window, right, looks much better, much lower attenuation. Low attenuation means the excitation photon can reach much, much deeper. That's what it means. That's a simple calculation. Of course, you do experiment, you can find out. These data are shown up by many groups. You can see it matches quite well. Experiment 800, experiment 1300, experiment 1700 nanometer. So the simple model really works out reasonably with brain tissue at least, right? So the best window are those long wavelength windows if you want to reduce excitation attenuation, the exponential functions. So let's hammer this water absorption issue sort of a little more. So what's the problem with water absorption? Again, blood absorption, water absorption. Okay, in vivo, so the curve looks like this, right? Again, typical 2 photons here. What we do is over there now, longer wavelength and 3 photons. So let's look at the comparison. Absorption, 1300, absorption, 900, 800, is a difference by a factor of 2. We do absorb here, factor of 2 more by the coefficient. But what happens is you win scattering by a factor of 2, right? Seems like, hey, it's a factor of 2 and a factor of 2. What's the big deal? The big deal is this. If you look at the math, absorption, too much absorption, what do? Reducing power, right? Reducing power of excitation. That compensates for the absorption. That's in the coefficient. You win scattering, as we talked about, that's in the exponent. So I always say, why this swings? You lose linearly, but you win exponentially. If you know any little math, you know exponential win is what you want, right? Compounding in a sense, if you do stock market, right? But in addition, in fact, the gain is even bigger because this is sort of a simple-minded analysis. What you need, because we're talking about brain tissue, photons don't go straight through the brain tissue. If they do, that'd be awesome, but they don't. They zigzag because of tissue scattering. When that happens, you have to use actually called effective absorption coefficient. It's a random walk calculation, which is well known in diffusive optics. You have to use this parameter. Essentially, you have to use the geometric mean between absorption and scattering. In that case, you can see you give up 2, you win 2. This number actually doesn't change. That means the effective absorption between this window and this window essentially are exactly the same, which experimentally also shows up to be the same. Now, over here, you give up maybe a factor of 10, a factor of 5 in absorption. You gain a factor of 3 in scattering. So this number is a little bit worse. But on the other hand, once again, you lose linearly, you win over here by a factor of 3 exponentially. So, and this shows very easily. So we started to do this about 10 years ago to show long wavelength really works. So we imaged the mouse brain vasculature by using 800 nanometer 2 photon and 1300 nanometer 2 photon. Very simple. Injecting 2 different dyes, fluorescing, excitable at the shorter wavelength, Alexa 680, excitable at the long wavelength. We just compare them, right? So you can see it's very easy to compare. At the surface of the vasculature, the image looks exactly the same. In fact, we make sure the photon numbers in signal are exactly the same over here and over there. Then all we do is just image deep. We just push the objective down, right, as we do 2 photon 800 and 2 photon 1300. You clearly see the vasculature we can image much deeper using longer wavelengths, right? Thai sci-fi laser typically 500-600 microns, you kind of top out. But this one you can see easily a millimeter. And in addition, it's a major advantage in overcome the shadow effect of blood vessels on the surface. You can see over here, lots of blood vessels. Over here, you can see kind of shadowed the dark areas by the top layer, large blood vessels. So it's going through the blood vessel much better, so the shadow is less. And you can see the movie of blood vessel structure. This is the depth, 400 by 400 micron field of view, going from surface all the way to about 1.6 millimeter into the mouse brain, in the gray matter area, in the frontal cortex, right? So it goes deeper and deeper, but now I want to focus on so-called contrast issues for 2 photons. You can see as you go deeper, blood vessels are still there, but the contrast gets worse, right? Inside and outside the blood vessel. You can see, you can kind of still see the blood vessel, but the contrast is getting worse and worse. And that turns out to be fundamental for the microscopy technique we have. Because you want to image that deep, that means what? All the excitation photons have to go through all the superficial layers on the top. And now, if you label blood vessels like this, all the blood vessels will be labeled, right? It's not like you only label something here. You label everything on the top. That means if you go deep enough, eventually all of these will give you a little bit weak excitation. But when you sum it up, it becomes very significant, okay? So this shows actually on this curve very nicely. You can see exponential decay, which is what we expect. As you go deep enough, you can see here it plateaus. In fact, you can go plateauing all the way to infinity. What happens is that if you go this deep, eventually the weak excitation, when they combine on the superficial layer, will actually overwhelm the signal generation that deep for 2-photon microscopy. And you can look at the contrast differences at the 1 millimeter, looks quite good. Black and white, that's what you want. But blood vessels labeled. You go deeper enough, you can see blood vessels kind of there, but outside is almost just as bright. Right? It's not lack of signal, by the way. We actually sit there, integrated this image for almost 4 minutes, just to make sure we have enough photons to show up. Right? So that's not lack of photons. It's just because the contrast is so low. Right? And theoretically, you can also estimate about 5 to 6 scattering length. You reach the fundamental limit for 2-photon microscopy in a uniformly stained sample like this. Okay? So that's, yeah.
Do you have it only at this depth, not before the surface excitation?
Yeah, you have it all the time, but at this depth, you kind of reach the absolute limit, means it's kind of overwhelm the focal point.
The curve is almost linear.
Yeah, the curve is good until that point, because even a contrast of factor of 2, the image looks great, actually. That's what it is. Because our eyes are so good to piece out this little bit contrast in the blood vessels. If you look at the last one, for example, like this one, you can still see the blood vessel, right? Even though the contrast essentially is 0. The pixel value difference is only 1, even after integrating for 4 minutes, right? So, but the eye is really good to sort of trace out those faint little things. You can still see that, right? So that's where the 3-photon coming in. So as we talk about this ability to form a 3-dimensionally confined focal volume is due to nonlinearity. So you can see 2-photon excitation without scattering in a log scale, right? This is my focal plane, let's see, right here. Away from the focus, the fluorescence goes down. This is like a line profile across the focal point like this, a line profile, right? Very localized. Here's a log scale, by the way. This is a million times lower than this. You go to 3-photon, sure, you go to much better confinement because it's high order nonlinearity. It goes down much steeper away from the focus. This Z is the distance away from the focus. So we know that for a long time, even 25 years ago as a grad student, I already know that. It's gonna have something to do. But we don't know what to do with this. We don't have any application. We don't have any context to think about this. The context when this becomes useful is when you have tissue scattering. Now, in addition to the geometric focus, like here, the focal point, you have this exponential function we talked about, the exponential decay function. So 2 profiles multiplied together. Now, in addition to the focal point, you have a lot of stuff above your focus because of this exponential function. So the background generation is the sum of all of those things, right? Now you can see if you go deep, this suppression of, say, 2 to 3 orders of magnitude becomes very important. Okay? Without scattering, you're comparing 0 to zeros. Essentially, you never see that. With tissue scattering, as you can see, now this becomes very useful. So now it turns out in this case, these 2 ideas, this long wavelength approach and 3-photon approach, is really very synergistic because if you want to use long wavelength right now to do 2-photon excitation, you basically don't have dyes to look at, particularly for in vivo imaging. If you think about fluorescent protein or some very good in vivo dyes, these wavelengths for 2 photon is just too long, right? This will need 850 nanometer 1 photon excitation. It's almost nothing. This will need a 650 nanometer excitation. There's a few you can do, but not many. If you add a photon, you can see, very nice. For 1300 nanometer windows, you can image blue and green fluorophores, green fluorescent protein, or GCAMPs. It just matches very well. If you go to orange and red fluorophores or fluorescent proteins, this is their 1 photon absorption peak. You multiply the wavelength by 3, you can see the number lands very nicely on the 1700 nanometer spectral window. So in this case, these two approaches are very synergistic, or very lucky in a sense. You have the 2 windows to match blue, green, orange, and red fluorophores. So kind of pretty naturally, you can combine those things. So the last issue we have to overcome to make it useful is this. The cross-section is really small. We still haven't addressed that. How do you make it useful? This is what I did in graduate school. As I told you, I know all those things in grad school, just don't know how to do it, how to make it useful. So we use a titanium sapphire laser. We imaged DAPI-stained DNA in a cell, no scattering, just all transparent single cell. We get nice images, 3-dimensional reconstruction, 50 microns, I don't know how many microns, just 3-dimensional reconstruction of this. But we actually have to use 50 milliwatts of power to get images like this. If you scale this for the mouse brain, you would need something like 5 watts if you want to image any significant depth in the mouse brain. If you do that, I think the mouse probably won't be happy, right? It's too much power you do. So the trick is very simple, actually. You have to use a different laser to drive the 3-photon process. And in 2-photon, what you use is a sapphire laser. The nonlinear signal actually is a function of laser duty cycle. The duty cycle is the repetition rate multiplied by the pulse width. Essentially is the fraction of time the laser is actually on. Makes sense? But the laser is mostly off. For a small fraction of time, they're turned on. So this number, for sapphire laser, you can see it's on 10 parts per million. That means 10 to the minus 5. For sapphire laser, only on for 10 parts per million, right? If you use lasers like this, that's what we did. You can see it's inefficient because you go to 3-photon, your signal is down by 1,000 times. That's why it's not practical if you use lasers like this. But now, because the 3-photon process goes as inverse squared of the laser duty cycle, that means if I reduce the laser duty cycle by using shorter pulses or lower repetition rate, if you do that with a few milliwatts at the sample, you can see the signal for 3-photon and 2-photon you typically get are very much comparable. So the key thing to realize to make it useful is that you have to use a different laser, probably at the few megahertz, but with more energetic pulses for 3-photon microscopy. Otherwise, it's sort of like you have a different engine for a different car kind of idea, right? You have to drive it differently. Of course, that's why it took about a few years to make it happen, because you look around, laser like this does not exist. So that's the great thing about my telecom background. Actually, we made the laser to make it happen. So that took extra time. So I'll skip all of those. So just to show that it's not just a mathematical analysis, it works in the experiment. So I compare 2-photon microscopy and 3-photon microscopy in the same field of view of the same mouse at the same time. Okay? The same detector, same objective, everything the same. This is 800 microns deep for blood vessel imaging with fluorescing dextran label. 2-photon at 920, 3-photon at 1300 nanometer. You can clearly see the difference. This is the blood vessel, this is the outside. For 3-photon, you can see the contrast, white and black. For 2-photon, you can clearly see the contrast is dramatically reduced. That's not electrical noise because the same detector is used. This is not dye leakage because if that's the case, it will be also bright over here, right? It's due to out of focus background when you have a uniformly labeled sample when you go deep enough. That's the logic, right? You can also look at a GCAM6 labeled neurons. This is a very densely labeled mouse. We actually want to label 100%. We haven't succeeded at it. We try to now go 75%. We'll see how well we can do. But this is very densely labeled. You can see for 2-photon microscopy, this is about 780 microns deep below the surface of the mouse. You can see the blood vessel should be totally dark, which is very dark in 3-photon images. In 2-photon, you can see it's not very dark because of the superficial layer contributes the background fluorescence. And also you can see a lot more neurons. You can even see nuclear exclusion, right? All the way down there. In two-photon microscopy, it's very hard to identify those things because the background goes up as you go deep. And we can quantify all those things. We can draw a line, for example, like this, right? Signal, background, right? You do a division. You can do SBR, signal to background ratio. For 3-photon in the blue, you can see it goes all the way very high, above 30, all the way down to hippocampus. In 2-photon, as you can see, as you go 700 to 800 microns deep, this SBR ratio, the SBR, really goes down to significantly less than 1. That means the contrast is much worse. Okay, we can quantify that. This is for fluorescing. These are for GCAM6 labeled neurons. These are for GCAM6 labeled neurons. These are for fluorescing. Okay, so clearly you can see the difference. Now, this is something might be surprising to you is that, signal is weak for 3-photon because it's a high order nonlinear excitation. But this I want to tell you is that if you go deep enough, the long wavelength advantage can actually overcome the weakness of 3-photon cross-section. And the reason is actually simple to understand. I'll give you a simple argument, then I'll show you the data. The signal goes as power to the nth power. 2-photon is 2, 3-photon is 3, right? Cubic and square. If you want to maintain signal as you go deep, what you have to do is that you have to exponentially increase the power at the surface because the excitation power goes down exponentially. Make sense? That means if you want to have the same amount of fluorescence coming back at you, you have to increase the power exponentially. Now that's where the long wavelength really helps you. Why? Because the exponential function has a denominator of effective attenuation length. For the longer wavelength, it's longer. For the shorter wavelength, it's shorter. That means one goes up much steeply than the other one. So eventually you can think about it. In the surface, 2-photon is better. If you go deep, 3-photon will catch up. That's the logic. And you can see the curve exactly like this. This is power required or pulse energy required on surface to give you a certain amount of fluorescence. At the surface, you can see 2 photons here, 3 photons there. That's why it's very obvious. If you want to do shallow imaging, do 2 photon very clearly. It wins all the time. Because you have to use so much more pulse energy to get the same signal back. But you can see 2-photon power increases much steeper if you want to go deep because of wavelength difference, right? You can see eventually they catch up just about 850 microns or so. That's for fluorescing, okay? If you go GCAM6, you can see the GCAM6 difference actually is smaller because the 3-photon peak for GCAM6 is exactly the 1300 nanometer, just by luck. Okay? For fluorescing, this is actually not the best wavelength to use. All right? So the projected crossover is also around 700 to 800 microns. So what does that tell you? That means if you want to do GCAM6 imaging, let's say that's what you want to do. That means, and you want to go deep beyond 700 to 800 microns deep, you got to do 3 photons 1300. There's nothing to lose. Even if you only label one neuron, you get a better contrast and you get a better signal with the same laser power injection. Right? So in that case, the changeover is roughly about 700 to 800 microns deep. We can also compare the red, orangey-red fluorophores. This is TD tomato labeled neuron, the mouse cerebellum. The cerebellum is actually a piece of brain that's actually tougher to image through. It's a folded structure with lots of fibers. So we just show that same comparison works. We haven't published that yet. We got the mouse from Utah, and this is in collaboration with Zella at Oxford. You can see that we can image much deeper at the 1700 compared with 1064 nanometer. Same comparison, same mouse, same time, same field of view, right? This is third harmonic generation, tells you the sort of like the myelin fibers in the mouse brain tissue. This is the neuron, and you can look at the blood vessel imaging too. This is Texas red labeled blood vessel in the cerebellum. And we go all the way down to here, which essentially lose contrast in the cerebellum, and you can see much deeper penetration for the blood vessel imaging. Once again, third harmonic generation, always the second channel you can look at. Again, same mouse, same time, and same field of view. In the cerebellum, actually very interesting because you have lots of these folded structures. You can see the decay curve. This is signal, this is depth, matched the third harmonic generation. You see the decay curve becomes a little bit chunkier instead of like a very nice linear exponential decay. You see the different decay slopes. And the reason is because every time you encounter those densely fiber region, your scattering is much higher. So the decay becomes steeper in those regions. So you have sort of a chunkier decay curve like this. Okay? We can image through the unthinned intact mouse skull. In this case, the bone is unthinned, and we just cut open the skin and look at through the bone, right? You can see the bone is over here, third harmonic generation of the osteocytes, the microvasculature all the way down to maybe 600 microns. That's through the intact, unthinned skull. And the movie shows you that. Osteocytes, the blue dots, or the green dots, third harmonic. You can see the blood vessel microvasculature, pretty clean, goes all the way down to about 600 to 700 microns. In this case, the bone is untouched, right? And then we can also look at neurons in this case, in addition to the vasculature through the bone. You can see the neurons, red fluorescent protein labeled neurons at about 200 to 400 microns deep, XY frames. We can image over multiple days to find the same neuron through the bone. Okay? Once again, the green are third harmonic, the red are the neurons. Okay? So these are unthinned skull. You can interrupt me anytime you want if you have questions. Okay?
What is the temporal resolution of your acquisition?
The resolution? Temporal resolution, these frames, I don't remember. I think normally we do about 4 to 10 seconds per frame. Because of repetition rate issues, yeah. But I will show you the activity imaging, then we speed it up because you have to look at a much higher frame rate. And again, we want to look for activity as we just transition over. And we use calcium indicators. The data I will show you is using GCAMP6 labeled neurons, either transgenic mouse or virus injected. We have also done some work with GR Gecko with 3 photon to 1700 nanometer. It doesn't look as good as this, but we still have some data of this at the hippocampus too, but I'm not going to show that today. So we have all shown work on the green stuff, GCAMP6 labeled. And we measure the cross-section of the GFP just to make sure the number we talked about is reasonable, and you can see it's within about a factor of 5-ish of what we expect. So the number is pretty boring because nothing new came out of those measurements, but the good thing about that is that we know we can actually saturate those fluorophores way before we reach the nonlinear breakdown threshold. Now that's very important because if you cannot do that, you might worry about nonlinear damage. I'll go back to this point in a few slides. What we first did is compare 2 photon, 3 photon recorded brain activity of the same neuron, just to show that no matter which modality you use, you get exactly the same activity trace. So in this case, it's spontaneous activity of one neuron doing 3 photon, 1300, 2 photon, 920 with GCAMP6. So you can see we put the 2 photon pulse in between the 3 photon pulses. We switch them back and forth within 1 microsecond. So that means in all practical purposes, these activities are recorded simultaneously. There's only 1 microsecond difference between this pulse and those pulses. Right? So you can see the activity traces look exactly the same. 2 photon in the purple, 3 photon in the blue. And we recorded over a couple hours for multiple neurons to show that the statistic is valid. It's essentially identical. And the delta F over F value actually turns out to be the same also. Okay, that's a little deeper than simple sort of a thinking, but we have some explanation for that. Anyway, but the bottom line is that the delta F over F are essentially the same for 2 and 3 photon microscopy. So this is image the layer 6 neuron.
Why should they be different?
Why this will be different?
Why should it be?
This is certainly expected, you would think, but this actually is not so obvious.
Why?
Because the delta F over F is obtained due to absorption spectrum change of the chromophores. If the difference due to quantum efficiency change, then there's no reason there will be any difference. But since the difference is derived from absorption spectrum change, as you know, in 2 photon, you go to 800 nanometer, you don't have any delta F over F. So the absorption spectrum actually changes when it binds calcium. That makes sense? If that's the case, then in principle, because these are 2 different physical processes, right? One is 2 photon, one is 3 photon. The absorption spectrum could be quite different. But there's a different explanation. It's a long paper. I'm still reading it. But I think there might be some explanation for that. But the bottom line is the measurement shows there's no difference. So this is the deepest layer 6 neurons because you see the third harmonic generation of the myelin fibers of the white matter. And we can see the activity. You can see the neuron kind of lines up with those fibers. They're sort of jammed into those fibers. These are the frame rate, field of view, pixel numbers, mouse age, and the power used. Okay? So that's what this is. And we have success rate in this case is less dependent on surgery. So we have essentially 100% success. Even the window is not perfect, you can still do this for the layer 6 neurons. And we can quantify the fluorescence in numbers of photons per frame. I think this actually is very useful because with the photon counts, we can actually ascertain these, we can actually differentiate noise from signal in a very quantitative manner, okay? Because we know how many photons we have. And these are very much short noise limited fluorescence acquisition. And of course, most people show delta F over F as a function of time. You can pick the neurons, you can see the delta F over F traces. But in traces like this, it's very hard to quantify, is this little peak a real peak or that's a noise peak? But with those traces, you can actually say with confidence, do you have 50% confidence or 95% confidence if you know the photon numbers? This imaging through the entire mouse cortex, cortical column, with 18 sections. As you can see the movie, we take 18 sections from surface all the way down to the deepest layer 6, about 800 microns deep. You can see the purple again. That's the, again, third harmonic of the myelin fiber. So we know we reached the bottom of layer 6, right? Bottom of layer 6. You can recall movies of the entire cortical column. And these are power we use, frame rate, and field of view. Now, we recorded this sequentially. But why do we still do this? The reason is because you can see that if you sum up all those power, that means you can actually have the power budget to record all of those simultaneously. If you can do the engineering to create those sections simultaneously and somehow discern those photons out, which could be done. And in addition, this is done with all 3 photon excitation. So in some sense, that's pretty dumb because we actually want to do 2 photons for all those sections because you can reach that with lower power and do the deeper layers with 3 photons because this is very densely labeled. You can see how green each frame is, right? The cells, the processes. So I think we actually have the power budget now to do something like this of the entire mouse cortical column simultaneously. So that's, of course, a lot of engineering left to do to make it happen. And we want to reach beyond the cortical layers, go through the white matter, reaching the hippocampus. And it turns out this layer called the white matter, it's called white matter because it scatters light a lot more. In fact, we can quantify this by looking at the fluorescence decay curve. Gray matter, white matter, and gray matter. You can see the white matter, it goes down much steeper because of more scattering in this very densely myelinated layer. Okay? So about 2.5 times as decay as this gray matter place. This shows you the SP layer imaging in the hippocampus. The purple, again, is the white matter using third harmonic generation of the myelin fibers. And neurons sort of in the deepest layer, 6, and some other neurons on the top. And this is the fly through the stack as a function of depth from 500 microns deep. Purple, third harmonic. Green are the GCAM6 labeled neurons. You can see the white matter coming through. See the white matter coming through. And for a little bit, about a millimeter or so, you can see the SP layer coming up, right? So that's the, and you can see a little bit processes too going further down. So there's actually some more things to look if you can go even deeper than this. And activity imaging in the SP layer, you can see that's just the XY frame, about a millimeter deep. And you can see the neuron flash. So watch mouse thinking very deeply over here. And we image the mouse multiple times over several weeks, try to find the exact same neuron to record. This is all spontaneous activity, by the way. We didn't stimulate the mouse in any way. It just, actually, it turns out in the hippocampus, the neurons are actually always quite active. In the cortex, actually, if the mouse is not stimulated, it's not nearly as active. Not sure any significance for that. Okay, and we can quantify with photon counts. It turns out you need roughly about 130 photons per second, or just about maybe 14, 15 photons in this frame rate to get about 95% confidence rate for those little peaks. This is the brightest neuron. This is the dimmest neuron, just to show the photon traces. And they can do, delta F over F traces like this, right? And just to emphasize, previously people imaged this layer through the white matter. You have to reach, you have to take the brain tissue away, right? Image this. Or what you can do is put a green lens, a greater index lens, or putting a transparent glass plug, push it down to image this layer. But now we can image this with the superficial layers non-disturbed, right? And the resolution is good. We take a cell, for example, like this. We draw a line, cross it. We can see the cytosol, the nuclear exclusion, like this, right? You can see the cell, dark in the middle and the cytosol on the edges. So the resolution is still quite good. In fact, we actually have better resolution data to show that the resolution is just about less than 1 micron, XY. The Z is about 3 to 4 microns. So what do we give up, right? We say it's great, we do 3 photon, we can long wavelengths, deep and others. You must give up something. There's no free lunch. You got to give up something to gain something. What do we give up? One is we have to use higher peak power, more pulse energy at the focus. If we do that, you worry about nonlinear damage. You got to be careful here, right? If you put enough pulse energy there, you can micro-machine the mouse brain. You can cut things away easily. We have done that accidentally, right? If you look at the situation, for 2 photon, typically you put 0.2 nanojoule at the pulse focus. That's 2 photon is enough. And you saturate the dye at about nanojoule. That means that for 2 photon, you never need more than 1 nanojoule at the focus. You put more, you're just killing the mouse and doing nothing else. For 3 photon, we actually use 1 to 2 nanojoule at the focus. And you saturate the dye roughly at 5 nanojoule. Now, the good thing about this saturation calculation is because we know the damage threshold typically is about 10 to 20 nanojoule. So we're still a ways away. So that's what you should do in the black arrow over here. We're still about 5 to 10 times away from the damage threshold. And this is very much a thresholding event. Usually you go 10 times lower, you have nothing happens. Okay, you can actually quantify that too. So that means you just have to be more careful. You don't want to accidentally put a lot more pulse energy to kill some blood vessels and the neurons if you put energy like this. And second thing you give up is that we use lower repetition rate. That means you cannot image really fast because you don't have enough laser pulses per second, right? And typically we use, I think this is roughly the optimum at the hippocampus, about 1 millimeter deep, you do 1 megahertz. That means you have 1 megapixels per second. You can use it to any way you want, right? Frame rate and the pixels per frame. You can compromise those 2 things. But that's roughly what you're going to get. If you do layer 6 neurons, you can put maybe 2 to 3 megahertz pulses because it's a bit shallower. You can put a little more pulse repetition rate. And again, you get 2 to 3 megapixels you get per second, and you put it anywhere you want it. And hopefully I convince you that for calcium imaging at least, these sampling rate actually is enough. That's the key. You cannot do voltage imaging for sure. But on the other hand, voltage imaging even with 2 photons is a bit too slow. Usually people use a camera to do voltage imaging. For calcium imaging, since these dyes are pretty slow anyway, these speeds are actually reasonable for it. How do we go forward? Next 5 minutes. Okay, the lasers are already here, which is great. You have a spectrophysics laser. You have the coherent laser. They are commercially available. They're reasonably robust. These are like I would say if you use the Tassafal laser in the 90s, these lasers are very similar to that. It's pretty good, but it's not perfect yet. They cover both spectral windows, which is very nice. It's widely tunable, computer controlled, and reasonably user-friendly. Okay? Basically means normally it works. If it doesn't work, you have to call the engineer because you can't touch anything. It's all sort of a sealed box, computer controlled. And I think we can improve the repetition rate of those lasers by another 2 to 3 times. And I think they can do it. It's not very hard to do. They are doing it, and hopefully it will come out within the next 6 months. So we can optimize the system fully. And we also adaptive optics. We're changing, borrow a page from astronomy. The brain is sort of a little bit heterogeneous. A perfect wavefront coming in gets distorted, and that happens, your focal point gets distorted. What you do is, if you can pre-compensate the waves coming in, you can actually cancel out the distortion caused by the brain tissue. That's the logic. Astronomy has been doing that for decades, looking at the stars in the sky through atmosphere. Now they are perfect with it. In astronomy, we are just starting in the brain stuff, right? But you can also do that. And the deeper you go, the more you want to do this. Because we do 3 photon, the high order nonlinearity, it turns out this is also becoming more important. So just preliminary data, haven't published yet. You can see we got a factor of 5 improvement at about 1.5 millimeter deep. This, I think, is the CA3 neurons in the hippocampus. We barely can see it. We're still working on this. You can see some neurons over here, very faint. All right, this is RFP labeled. RFP actually turns out to be dimmer than TD tomato. If you can label with TD tomato, you probably get a lot better data than this. So one thing we do realize is that when you go to longer wavelength, actually the aberration is naturally reduced because physically what happens for aberration is a phase error in your wavefront. It goes as path length difference divided by lambda. It's very simple mathematics. If the wavelength is longer, your phase error is just naturally smaller. So in the sense that we do need it, but you can see the gain is not as big as I hoped, like 20, 30 times. It's only about a factor of 5 at the maximum. That's what we have seen so far.
Sorry, that was with the bone or without the bone?
That's without the bone. Yes, with the cranial window. Yeah. So of course we want to push into biology and neuroscience. So laser is there, a little more expensive still. Long wavelength optics, which is great now, is all available from a variety of microscope companies. You just have to have the money to buy it. It's there. A microscope is now hard to make. So all these are there. And we need fearless biologists who can try this to do some real biology with. Right? Otherwise, I can quantify, I can demonstrate, but there's not much biology coming from my lab. Right? And the good thing is there's already 10 to 20 groups have either set it up already or they're hopefully soon to set it up to do some real science with. Because only the real science will open this field up. Because people usually, biologists look at me always a little bit suspicious, like, is that really right? Right? If you can do real biology, that changes it. And so you can close the loop between instrument people like me and the neuroscience that advances. So that's, I think, is what the next step would be. In fact, I think I need to get out of this field already because I'm done. So, okay, to conclude, should we abandon 2 photons? I hope to convince you no. If you just want to do this, 500, 600 microns deep, 2 photons is better. Lower power, higher speed. Of course, why not? But this technology, we developed 3 photons at those long wavelength windows. Has advantages. If you're going to go really deep, right? For example, mouse hippocampus, I showed you. Deep layer of the rat and the primate brain, very useful. If it's really densely labeled, all the neurons are labeled, for example, then maybe even for layer 5, layer 6 neurons, you want to do this because of background generation on the superficial layers. That's for densely labeled brain. And lastly, if you want to overcome a highly scattering layer, such as like mouse skull, the bone, we're imaging spinal cord now through the white matter, actually has some reasonable success. Imaging through dura, Timo is very interested in. And I have data to show, and other people have demonstrated already, we can image through intact fly cuticle. Sorry, this is misspelled, I guess. I'm not sure. So that actually opened up new neuroscience for fly work because the fly can now live forever, in a sense, but the cuticle is intact. So we can actually definitely do that. So thank you very much.